The knowledge of amino acids is critical for the understanding of many additional topics in biochemistry, including protein structure and protein function. There are three main functions of amino acids. The first is that they are building blocks of proteins. Secondly, amino acids are the precursors of neurotransmitters, such as dopamine shown below in the figure, which is derived from the amino acid tyrosine. Third, there are a variety of other functions in the cell, such as citrulline, which is the structure shown on the right-hand side of the screen. The general structure of an amino acid is that the amino group is on the alpha carbon relative to the carboxylic acid group. In addition, the alpha carbon always contains a hydrogen atom, and it contains an R group of some kind. This structure is incorrect in a very important way that we will go into shortly. A concept from organic chemistry comes into play here. That is, that unless the R group is either a carboxylic acid group, which it never is, an amino group, which it never is, or a hydrogen, which it is in one case, the alpha carbon is a chiral carbon. There are 4 different groups attached to that central carbon. What this means is that the amino acid can exist in 2 different forms, which are shown on this slide. This type of structure is referred to as a Fisher projection, and very importantly, the groups that are extending left and right are pointing out towards you as an observer. I am drawing my structures differently than those in your book, and you should go through using a model kit and verify that in fact they are the same structure. The 2 forms, one with the amino group extending towards the left, is referred to as the L amino acid, and the other with the amino group extending towards the right is the D amino acid. The form that is physiologically relevant is the L isomer, except for some unusual situations such as the peptidoglycan layer of bacteria. Note that again, both of these structures are incorrect in a way that we will now address. The incorrectness of these structures is that at physiological pH of 7.4 is above the pK value for the carboxylic acid group, and therefore the carboxylic acid group is negatively charged. pH 7.4 is beneath the pK value of the amino group, and therefore the amino group is positively charged. This form of the molecule is known as a zwitterion, and I would characterize as meaning both at the same time. That is, there is a positive charge and a negative charge, but the net charge is 0. As you memorize the structure of the amino acids, you should memorize in this form. A few properties of amino acid include that, because of the positive and negative charge, they are by definition ionic compounds and therefore have a high melting point and are solid at room temperature. The amino acids having these charges are for the most part very water soluble. There are some exceptions that we will discover in the laboratory. Also, the average molecular weight of the amino acids is 110. This is an important tidbit for calculating the approximate molecular weight of proteins and represents one of the end of chapter problems that is assigned. You should, as biochemistry students, commit to memory the structure of the amino acids at physiological pH, the 3-letter and 1-letter abbreviation of the 20 standard amino acids. You should also be thinking, but not memorize, the various classifications of amino acids, which is whether they are polar or nonpolar, what type of functional group they have, whether they are acidic or basic or neutral as amino acids. In an on-ground course, I spend an entire 50-minute class period drawing out the structure of all 20 amino acids, including throwing in some tidbits of modifications that you will find in proteins and other characteristics of the amino acid. But that is not a practical use of time in an online course. Do not let the absence of an hour-long lecture trick you into thinking that the structures are not important. They are of vital importance. Among the chemistry of the amino acids, acid-base chemistry is one of the most important. In this slide, I have drawn the 3 possible structures of the general form of an amino acid at various pH values, 1, 7, and 13. At pH of 1 or some other very acidic pH, the Zwitter-ion is not present because we are beneath the pK value of the carboxylic acid group. Each amino acid has a characteristic pK value, but the pK value of the carboxylic acid groups are all in the range of about 2. At pH 13 or some other very basic pH, the Zwitter-ion is not present, as both the carboxylic acid and the amino groups have both been deprotonated. The triangles that are descending in height on the bottom and increasing in height on the top represents the fact that as we move from pH 1 to pH 7 to pH 13 by the addition of base, the number of molecules of the left structure is decreasing, and the number of molecules of the Zwitter-ion structure are increasing as we approach pH 7. As we go from pH 7 to a basic pH, the number of Zwitter-ion molecules will decrease, and the number of molecules that are on the right-hand structure will be increasing. Thus, the possible structures would exist at pH 1, physiological pH, and pH 13. We can call A, B, and C for reasons that will become apparent momentarily. The next question we want to consider is, what would a titration curve of this amino acid look like? What are pH values at the inflection points, and what species are present at each? This slide is a titration curve I collected for use in teaching biochemistry. It is the titration curve of glycine by adding drops of 0.1 molar sodium hydroxide and measuring the pH after the addition of each drop of base. At pH 2 or so, which I have indicated with a green dot, the structure of the molecules that would be present is 100% of the A form of glycine. The black dot at the vertical inflection point would represent where we have 100% Zwitter-ion present, and the blue dot, representing the pH as high as I could get, would represent the place where we would have 100% of the C form of the molecule. In red, I have approximated the horizontal inflection point representing where there is buffering occurring. Remember from our last video that buffering occurs when the pH is equal to the pK value and there are equal amounts of the A- and HA forms of the molecule. In this case, that point then represents where we would have 50% of the structure A and 50% of the B Zwitterion structure. There is another inflection point, not as apparent in this graph, which represents the pH equaling the pK of the amino group and represents the place where we will have 50% Zwitterion and 50% of the C structure. I did not actually put a dot for that on the slide. The vertical inflection point represents a very important point referred to as the isoelectric point, PI. The PI, which is defined as the pH at which the net charge is 0, is calculated mathematically by summing the pK value for the carboxylic acid group and the amino group and dividing by 2. Table 5.1 of your book lists the pK value, and for glycine we will be adding 2.2 plus 9.7, which equals 11.9, and dividing by 2, which is going to be a PI of 5.95. All of the neutral amino acids, of which there are 15, can be determined in the same manner. There are 2 amino acids, aspartic acid and glutamic acid, that are acidic amino acids. If we ask the same questions as we did before, what are the possible structures? What we will come up with is as follows. At a pH of 1, structure A will exist. At pH 7, the structure in the middle will exist. And at pH 13, the structure on the right-hand portion of the slide will exist. But this skips going over very methodically the possible structures. We really need to figure out, as we deprotonate through pH 7 and 13, exactly which proton is going to be attacked by base first. To answer that question, we consult table 5.1 of your book and find that the pK value of the carboxylic acid group for the amino acid glutamic acid is 2.19, which your book calls 2.2. The pKa for the R group is 4.25. Therefore, at a pH of 2.19, and I'm going to put that in quotation marks for later on, the structure that will exist is in the lower left portion of your slide in green. We will call this structure B. As we increase the base addition, the next group to be attacked will be the R group of the amino acid, and we derive a structure which we will call structure C. If we want to consider the question of what is the PI, We have a bit of a question on our plate. In the case of glycine, we simply added the pK value of the carboxylic acid group, the pK of the amino group, and divided by 2. But in this case, should we add 4.25 and 2.19? Or should we add 4.25 and 9.67? Or should we add 2.19 and 9.67? To answer this question, we need to go back and think about what is the definition of Pi and the net charge of each of these molecules. So for structure A, I see one positive charge and no negative charges, so the net charge is a +1. And I've written that in the kind of the fourth line down on the slide. In the same manner, when we look at structure B, I see that this is the Zwitterion form. Structure C has 2 negative charges and 1 positive charge for a net charge of -1. And structure D has 2 negative charges and no positive charge for a net charge of -2. Now we can consider the question. The Pi is always going to be the sum of the pK values around the Zwitterion form divided by 2. The Pi, consulting a table of pK values, is 2.19 plus 4.25 divided by 2, or 3.22. I will also calculate and put in parentheses a value which results in 6.96, and this will become relevant on the following slide. Here we have an actual titration curve of glutamic acid presented in a slightly different form than my last one. What I have done here is to place some red dots along our titration curve. Recall that we calculated that we should have a Pi value of 3.22, and the second red dot from the left approximates, but looks a little bit higher than 3.22. We also calculated a value of 6.96. This is approximately our horizontal inflection point. Because our pK values are 2.19, 4.25, and 9.67, we should see the first, third, and fifth red dots are approximately at those values. To test your understanding of this, I will have you go through the same process of drawing possible structures, determining net charge of each of these structures, consulting a table of pK values in order to determine where inflection points, including the Pi, should be observed, and sketching the resulting graph.